Mhm. Today we are going to discuss about the next generation sequencing. This lecture will only cover the theoretical aspect of next generation sequencing, and this video will be followed by certain sub lectures that will cover the practical aspect of the next generation sequencing. For example, if someone has the next generation sequencing data, how we can analyze the data by using the Linux platform, that we are going to learn in the subsequent sub video lectures of this topic. So let's say if a person is coming in front of you and is coming just with the DNA or the RNA sample, then what is going to be the first step in case of that if your sample is DNA or if your sample is RNA? So the first step is going to be the library construction. If your sample is DNA, so you will prepare the DNA library. If your sample is RNA, you will prepare the RNA library. For example, if your sample is genomic DNA, so in that case, because you know the size of the genomic DNA is 3000 megabase pairs, so first of all you have to convert this genomic DNA to the fragmented DNA, and that you can use do it by using certain enzymes, for example DNase. Let's say if the quantity of your genomic DNA is very less, in that case you have to do the PCR. In the PCR you will use certain primers, forward and reverse primers, and those primers will bind to certain portions of this genomic DNA, and based on that you are going to get the amplicons. So if your genomic quantity of the genomic DNA is less, you can do the PCR. If the quantity you have sufficient quantity, then you can directly fragment the genomic DNA to the fragmented DNA by using certain enzymes, for example DNase. After you have the fragmented DNA or the amplicons, you can ligate these fragmented DNA or the amplicons with the adaptase molecule. What are these adapter molecules? We are going to learn in this lecture only later on. Let's say if your sample is RNA, whatever your sample is, you have to fragment those samples, and it is because Illumina sequencer has some capacity, limiting capacity. It cannot measure the sequencing reads if it is more than 600 or 300 base pair long. So let's say if your RNA is 10,000 base pair long or 1000 base pair long, then it cannot sequence it. So therefore it is necessary to fragment these RNA molecules. So once you are having the fragmented RNA molecule, you can directly convert to the cDNA molecules, and then you can ligate these cDNA molecules with the adapters. Or what else you can do, you can directly ligate these fragmented RNA molecules with the adapters, and then you can convert that to the cDNA. So either way you can do. The next one more method is there in which you can convert this whole RNA molecule to the cDNA, and then you can fragment the cDNA molecule and then ligate them with the ligator molecule. That you can do by using transposon 5, TN5, but the that process is very not very reliable, because in that case you need to know what should be the ratio of your RNA and the TN5, so that you need to standardize. When you need to standardize that, when you are having the, let's say your full length RNA, you convert that to cDNA. Let's say if this is RNA, you have converted to cDNA, and then you are fragmented this cDNA. Okay, in that case you can use the TN5. So what TN5 will do, it will fragment your cDNA, at the same time it will ligate the adapter molecule to the cDNA molecule. So this is not very frequently used method in which you are directly converting the intact RNA to the intact cDNA and then you are fragmenting the cDNA. The most usual approach is to fragment the RNA molecules, and then you can ligate the ligator molecules at any step after you are making the cDNA, for example here. Then you can ligate the cDNA with the adapter molecules, or you can directly ligate the adapter molecules to the RNA and then you can do the RT-PCR to have the cDNA. So this is all about first step. What you are going to do if your client is having the DNA, or if your client is providing you the DNA or the RNA sample. First step is going to make the library, and that you can do by using some Illumina kits of library preparation. Is less than half an hour job. You can do that. So let's say in this we said you have the DNA fragmented molecules and then you are ligating these molecules with the adapters. So what happened if you have the DNA fragments? All of your DNA fragments are not going to be the blunt ended. It may be blunt ended, it may be overhanged, 5' overhang or 3' overhang. As you can see here, it is a 3' overhang. So first step, after you are having your DNA fragments, you need to treat your DNA fragments with certain enzymes which will remove this overhanging portion and you will get the blunt-ended DNA molecule. After you have these blunt-ended DNA molecules, the next step is going to attach the adenine base pair at the 3' position of your DNA fragment. And why we are doing it? Because your adapter molecule contains 5T at the 5' position, so it is easy to ligate the adapter molecules with this DNA molecule, blunt-ended molecule. For example, here you can see there will be T. For example, here it will be T in the adapter molecule, where this T will be in the adapter molecules. So that's how you can do the library preparation. Now we will understand what is actually the adapter molecule. So the adapter molecule will have your fragmented portion of your DNA or RNA or cDNA, sorry. Then you will, in the single index molecule, it will be having the one barcode sequence. Barcode sequence is usually used so that the sequencer can identify which sample is coming from which part. For example, you are having a mixture of samples. Some are like diseased samples, some are the healthier samples. Then your healthier samples may be labeled with the I7 index. Your diseased samples can be labeled with some other barcode sequence. So the sequencer can identify this sequence is coming from the healthy person and this sequence is coming from the diseased person. So these are the index molecules. These are not the labeled molecule. These are just the unique sequences that is attached to the adapter molecules, so that the sequencer can easily identify the identity of your sample, whether it is diseased, whether it is treated, or some other things. You can see whether it is mutated or whatever, based on your experiment. So this is your DNA fragment, which is the insert. Then you have in the single index molecule, you have only the single barcode sequence. So the sensitivity is little bit lesser. Then you have the P5 and the P7. These are the platform specific sequences for the library binding to instrument, so that we will describe it in a short while. And then you have the SP1, which is the sequencing primer site. So these are the sequencing primer site. So what is your adapter molecule? You can easily see they have the sequencer that can bind to the flow cell of the sequencer, which is P5 and P7. These are all sequences only, nothing else. Okay. Then these are the sequencing primer site, and you have in a single index you have a single barcode sequence. What happened in case of the dual index, which is unique dual index, commonly known as the UDI? you have the two indexes, I5 and the I7 indexes. So you are providing two unique sequences to your sample to be very much sure that your sample is what is the source of your sample. In case of the dual index UMI, you are having the UMI, which is the molecular index barcode, that is again to provide more sensitivity to your assay. The sequencer is quite confirmed that the particular sequence is coming from a which source or which sample. So just to provide more sensitivity, these sequences are attached. UMI-A is most commonly used in the Novasec sequencing. So here you can see the flow cells. So this is the high-sec flow cell. High-sec flow cell has 8, you can say the 8 columns. In each column you can run 12 samples. So how many samples you can run? If in this you can run, let's say 12 samples, and there are 8 columns, so how many samples you can run? You can run 12 into 8, which is 96 samples per flow cell. Okay, so this is your flow cell. And in a sequencer you can run this kind of 2 flow cells. So 96 multiplied by 2 which will be 192 samples you can run. Similarly, in case of the Novasec flow cell, you can run multiple samples. You can see there are 4 columns, and then these columns are again divided into multiple sub columns. So this is a Novasec flow cell. The read size you can see is a bit different. Here it is 3 billion reads it can read. Here it can 13 billion reads it can read. And read is nothing, just a 300 to 600 base pair sequences. These sequences are known as the read sequences. So what happened? This is what? This is the flow cell. What is this? This is your flow cell. Or you can say the platform. And this platform has multiple sequences. Let's say this is P5. This is P7. These are not labeled, just remember that. Just to provide you the clarity, Illumina has provided some coloring to them, but these are the sequences only. Now your P5 and P7 sequences, and the same sequences as you can see here, P5 and P7 again is in your adapter molecules. So let's say this is complementary sequence to P5. And this is, let's say, complementary sequence to P7. Okay, so now you can see this is P5. Then we need to say maybe we need to say this is P5 and this is going to be P7. Okay, and again I want to make it very clear, these are not labeled blue color or purple color, just to provide the clarity. Illumina has done that. So these are basically the complementary sequences. So for example, this is P7 and this is P5. So this blue color is P7 and the purple color it is P5. So your P5 is this portion which is complementary to this sequence is going to attach here from your DNA fragment, because this is your DNA fragment that contain the P5 and P7 sequences that are complementary to the P5 and P7 sequences of the flow cell. So now they will attach, and now the DNA polymerase will start adding nucleotides, and you will be having the second strand molecule. This one. Now you can see here, because this is attached to the you can see here, this is this white portion you can see here. This portion, okay. So this is basically, this portion is your platform. So this sequence, this particular sequence is attached to the platform, okay. And your sequence is not attached to the platform. So what will happen once the second strand synthesis is going to be completed, in the next step, washing step, your strand is going to be removed, your fragmented DNA strand, because this is not attached to the platform. Now again you will be having this sequence, which is the P5, then your sequence, and then P7 sequence, and this is attached to the platform, and therefore this sequence is removed because it was not attached. Now it will form the cluster. It will go and bind to the P7 part of the flow cell, and then again the DNA polymerase will start adding the nucleotides, and the second strand in synthesis is going to be completed, and you will get something like that. And then again the process will again this will bind to the P7, this will bind to the P5, because this is purple colored, which is P5, this is blue colored, which is P7. So this P7 will bind to the P7 complementary sequence, and P5 will bind to the P5 complementary sequence, and again you will get the cluster generation. So in the sequencing, two things are going to be done. One is the cluster generation, which you are doing in this flow cell. Another one is the sequencing part, in which the sequencer will sequence these cluster molecules. So first part is to generate the cluster molecules. So now you can understand how the cluster molecules are generated, because the flow cells are having the P5 and the P7 sequences, and your DNA is again having the P5 and P7 sequences. So then it will attach to the flow cell, and then with the help of DNA polymerase, this second strand is complementary strand is going to be synthesized here, and after the washing step this part is going to be removed, and again this strand is going to be there itself on the platform. Now this is, because this is a P7 sequence, this P7 will bind to the sequence which is complementary to the P7, and then that's how the cluster will be generated. I hope it is clear now to you. And after the cluster generation process is completed, then the sequencer will sequence these sequences. So now this is known as the sequencing by synthesis. It's known as the SBS process. So you can see here, this is for example your clusters have been generated. Now what happened, now your sequencer will start adding the sequencing. This is the sequencing primer site, and you will be having the sequencing primers in your sequencing reaction that will bind to the sequencing primer site, and then the nucleotides will be added, and each nucleotide will be attached to the terminator molecules. So this is the strand by synthesis process. For example, if it is A here, then the T will be added. If suppose all 4 nucleotides will be added to this reaction, so only the T will bind to A. C, G and A will not bind to the A sequence, only the T is going to bind to A. Once the binding is completed, the terminator molecule is going to be removed, and then the next process will start, and again the 4 bases will be added. And because once again it is A here, so only the T will bind here, and once the process is there, the fluorescence is generated, and once the fluorescence is generated, that part is going to be captured by the sequencer, and then the terminator molecule. So this particular T, if you can see here, it will be attached to the the terminator molecule. So this is, you can say, the terminator molecule. So once T binds to A, then the fluorescence will be generated, and then this terminator is going to be removed. Then all the 4 nucleotides will be added, and once the T will bind to A, again the terminator is going to be removed, and then only the A will bind to T. So this is known as the sequencing by synthesis. And once this is done, this particular sequence is going to be removed because this is not bind to the platform, and this will be still attached to your platform. So that's how the sequencer is going to read the sequences. This process is known as the sequencing by synthesis because sequencing is done during the synthesis process. One more thing you need to remember, all the 4 nucleotides are attached to a single dye. They are not attached to the different dyes as in the Senger's method. The only difference is this: all the 4 nucleotides are attached with the terminator molecule. And once the binding is there, then only the fluorescence is emitted and the sequencer reads that particular base, and then the terminator is removed, and then again the 4 bases are added. But all the 4 bases are only labeled with a single dye. They are not labeled with a different colored dyes. So that is the difference between the sequencing of next generation sequencing and the sequencing of the Sanger sequencing. So now one more thing which I want to clarify here, as you can see here, this is the flow cell, this portion. This portion is your flow cell. Okay, you can see here they have provided the green color. As I already said, these are only sequencing, no color is provided to them. Just to provide the clarity only, they are providing certain colors to these sequences, but actually these are only sequences. Only the 5' portion is attached to the cell. As you can see here, whether it is P5 or P7, both are attached to the at the 5' portion only. That part you have to be very clear. So as you can see here, so this is your 5'. So let's say this is your flow cell. So this is your P5 and this is your P7. Okay, but the 5' portion only will attach to the platform. That part you need to remember. Okay, so what happened? Now the same process will start. Your strand will bind to this particular sequence, and this is going to be generated. Then the complementary strand to your fragmented DNA is going to be generated here, as you can see here, and similarly to here. This portion you can see, this one. This one, this and this. Just focus on this and this only, because this portion is not attached to your flow cell. This will be removed, as I said here. This is not attached to the flow platform, so this will be removed. Okay, so you have P5 sequence, then the P7 sequences. In the read one, what we are going to do, the sequencer is going to cleave the P5 sequence, so only the P7 is there. So in the sequence, then the sequencing primers will bind to the sequencing primer site, because you know in your adapter molecule, if you see, there is the sp1 and sp2, which is the sequencing primer site. So your sequencer will add the sequencing primers that will bind to the sequencing primers, and then the sequencing will be started. So this is the read one process. In the read two, what happened? Similarly, you just concern about this sequence. In this, what happened in the read two, you can see here these two portions, like here is going to be there, but then you are going to cleave the P7 sequence, not the P5. So once the P7 is going to be cleaved, then because the orange color is P7, so now we are cleaving the P7. Here we have cleaved the green portion, which is P5. Now you can read the other part of the sequence. So that's how the read 1 and read 2 are generated. Now one more thing, you have seen always that besides providing the sequencing primers, in the sequencing the index primers are also used. Why it is like this? Because sequencing primer can also read this index sequences, because index sequencing are just the sequences only that are nothing different than these insert sequences. These index sequences will also contain the same nucleotides A, T, C, G. Then why does this sequencing primer cannot read them, and why we are providing them the index primers? It is because the sequencing primers can read only up to 300 to 600 base pairs. If suppose your index sequences is far from the 600 base pair, then the sequencing primer cannot read that sequence. This is the reason the indexing primers are provided, so that the index primers will bind to this indexing primer side, and then the sequencing will be there. Then the sequencer will read the sequence. And again, see, this is the sequence by synthesis process, SBS process. So this sequencing portion is going to be removed only, because it's not attached to the platform. Only this is there. So whatever the index primers will read, that will recognize particular sequences from which samples. So in your reaction, you are providing the sequencing primers and the indexing primers, and based on that they will understand which sample is what. Here you can see the read 1 primer, you can see the DNA insert, this P7 adapter. Now you are adding the I7 primers that will bind to the I7 index sequences, and then they will recognize what is the sequences, because these are very nearby, and all the sequences will be added after the sequencing primers sequencing is done. So they will recognize which sample is coming from which source. Therefore we are providing the two primers. One is the sequencing primer, one is the index primer, because sequencing primer cannot read the sequences if your index is, let's say, more than 600 base pair away. And similarly it will go to the read 2. Now we will learn about so this is all about the next generation sequencing, how the clusters are formed, and then how the sequencing is done. And once the sequencing is done, you need to add the indexing primers to see what is your sample, what is the source of your sample, and once this is done, your sample and sample library part and the sequencing part in the laboratory is done. So next we are going to learn about the enrichment strategies, because let's say if the client is coming and say, I'm only interested in this portion, I'm not interested in this portion, then it's a wastage of your time to make the full length library. So what you will do, you will use some probes which are complementary to this sequence and attached to some beads. So these beads will pull out this particular portion in which the client is interested. So this is known as the enrichment strategies. Similarly, in case of the RNA, because RNA you need to deplete, so either you can use the because mRNA is attached with the contain the poly A tail, so you can use the poly T sequences which are attached to beads. So in this way you can enrich your mRNA, or you can deplete your rRNA by using similarly the beads like sequences which bind to the rRNA. So you need to enrich your mRNA molecule first. Okay. Now we will learn about the index hopping. What is index hopping? And here we can see easily when you are you have seen here in this is your insert sequence, and you are attaching to this adapter molecule. And when you are doing that these things, you may do in the 96 well plates, so what will happen if you are doing in the 96 well plate, and if let's say you have the one sample, this is your second sample, this is your third sample, this is your fourth sample, and then you will attach this sample with the I5 and the I7 sequence. Okay, so here you can see so this is your I5 indexes and this is your I7 indexes. Let's say if your I5 sequences and I7 sequences are different, what will be happened here? If you go down the column, so your I5 indexes, I5 sequences are going to be different, but your I7 sequence is going to be same. If you go across row, your I7 indexes are going to be different, but your I5 sequence is going to be same. You can see here, this is the magenta colored or brown colored, but the I7 index is going to be different. And again, these are not colored, these are just the sequences. So what will happen if this particular sequence leach from this well to this well, and let's say if in this well you have added the control sample, in this well you have added your diseased sample, so what will happen? This particular sequence, if this sequence is going to be leached from this portion to here, so you will get what you will get. You will get the I5 is going to be the magenta colored only, but your I7, it can be green colored, it can be this colored. I think this is magenta. You can see this is brown colored. So let's say if your sample, if this is going to be leaved from well number 2 to well number 1, the I7 sequences, what will happen? So your sample number 1, let's say if this is from your control sample, it will have the I5 sequences which is brown colored at 5 prime. At the same time, at the 3 prime, it may contain the green colored I7 sequences, but because since 2 is leached from well number 2 to 1, it can contain the magenta color sequences as well. So that is known as the index hopping. Index hopping can happen at any step of your sequencer. In the sequencing machine itself, it can happen while you are attaching your adapter molecule to the DNA molecule, then also it can happen because leakage can happen anyway. So this is known as the index hopping, where the sequencer is confused whether your sample is from disease sample or is from control samples. So it will not filter it out, it will be confused. Okay, it will be misassigned sometimes. It's rarely, but sometimes it happens. And let's say if your particular sequences are very important to you, it is a rare variant, then you don't want to take a chance. In that case, what you can do, you can attach the same sequences. Like in this, you can attach to instead of attaching 2 different sequences, you can attach the same sequences at the 2 sides. So here you can see P5 and P7 is of same color, so same sequences you are attaching. So even if it is leaking 2 to 1, you can easily understand, okay, this is not this sample. So this is just to increase the sensitivity of the assay. Sometimes unique dual indexes which are used, and they are having the same sequences for the P5 and the P7. Oh, sorry, I5 and the I7. So this is the here they are showing if they are of different color, but if they are of unique sequences, then what will happen? 98% chances are there it is going to be correctly read. If there are any misalignment, then it is going to be filtered out. Okay. Okay. So this is known as the index hopping, and it can happen at any position. So for example, if you are having the samples, multiple samples, you will fragment these samples, and then you will load here, and then this may be your control sample, this may be your disease sample, then this one may be treated with some dose, 1 dose, 2 dose, 3 dose, whatever the dose is, and based on that you will just provide them the sequences at the I5, and you will provide them the barcode sequences at the I5 and the I7 part. So either you can use the different sequences or you can use the same sequences. If you are using the same sequences, the chances of contamination of or the index hopping is comparatively less. Then this is just the advantages of Illumina sequencing. The market is 90%, and the imaging based method, because we are using the dye, which is single colored dye that is attached to the terminator molecule. It can read up to millions to billions per run. 300 to 600 bases per read is there. High fidelity, high accuracy, and it's a much faster process. So less than you can complete the whole human genome in just 48 hours, so it's a bit faster. So this is all about the theoretical aspect of this next generation sequencing, in which you should understand what is the sample crosstalk, which is the index hopping. You can see here what is the sample crosstalk and why it happened. You can understand easily in this example here. So that's all about the next generation sequencing theoretical aspect, and in the next lecture we will learn about the practical aspect of next generation sequencing. Thank you.